Hi, everyone. Welcome to one more of our virtual workshop of the Behavior Forum. So the Behavior Forum, as you know, is a collaborative place that is made by scientists for scientists to discuss animal behavior. This is how the Behavior Forum looks like, but you're all very familiar with this forum. But just to make a little reminder, this is a place where once you've entered, you can find the category you are interested in, and then you can just start interacting with peers in a very in a scientific way, but also in a very collegial and fun way. As a spin-off of our forum, we are organizing online workshops that we call Understanding Behavior. And it always happens on Thursday at 12:30 CET. And what is nice is that for these workshops, you can suggest speakers. You can also suggest yourself as a speaker. And if you're interested to do this, you can go on the forum or on our LinkedIn to get more information. So what is interesting with this workshop is that before and after the talk of our speaker, you can actually keep interacting with the speaker, ask questions, and get to know more about the topic discussed. So please don't hesitate to go on the forum, but also share that on LinkedIn if you're excited about it. So now, without further ado, I'm going to leave the virtual mic to Davor, who is the co-host with me today, and to introduce Megan to you.
Hi, everyone. Thank you, Marion, for a nice overview of the forum and what we do. And today we're very, very pleased to have Megan Jackson here with us from the UK. So Megan Jackson is a senior research associate at the University of Bristol, and her research utilizes ethological foraging behavior in mice to study behavior changes in psychiatric disorders, using a combination of tools, including pharmacological manipulations, phenotypic models, and freely moving neuronal recording. Megan aims to understand the pathways involved in intrinsic motivation and how it might help us understand and treat motivational disorders such as apathy syndrome. In my opinion, this is a very cool and relevant topic that can indeed very much facilitate translational research and can help us come about with some really cool knowledge in terms of specifically, I think the behavioral perspective that in the way that Megan is researching is really what we could use a lot more of. So thank you, Megan, and the floor is yours.
Great, thank you. I'll just share my screen. Okay, hopefully everyone can see that okay?
Yep.
Cool.
Looks nice. Yeah.
Great. So thank you very much for inviting me to speak at this forum today. So today I'm going to talk a bit about a new task that I've developed that really draws on species-specific behavior, the mouse's innate drive to forage for nest material. And I'll talk a bit about how we can use that behavior in a behavioral assay and understand more about motivational disorders and motivated behavior just more generally. Because this talk is generally about motivation, I thought it was important to talk a bit about apathy syndrome, because this is a really profound motivational disorder and really puts why we look at motivation in perspective. So apathy syndrome is defined as a quantitative reduction in motivation, or when we kind of get operational about it, a reduction in goal-directed behavior. And this definition is really useful for us because it's something that we can measure in both humans and animals. But by kind of summarizing it as just a sort of reduction in motivation, we start to kind of miss the multi-dimensional aspect of it. So I've highlighted it here to show that apathy is sort of comprised of a few different types of behavioral disruption. So we have things like behavioral cognitive disruption, so things like reduced effort to start and finish a task, emotional affective, so this could be something like an emotional blunting where you lack a reaction to something positive or negative, so something like a piece of news. And finally, social motivation, where you really lose that drive to initiate social contact. And apathy is really prevalent across a whole range of different neurodegenerative and neuropsychiatric diseases. It sort of pops up a lot in a lot of different places. And it's also really common in things like healthy aging and even stroke and infection. But of course, apathy isn't really talked about as much because things like depression and schizophrenia really dominate the field. But apathy really does have a profound impact, not just on the quality of life of the patient, but also their caregivers as well, because it's really sort of disturbing to see the person you love really start to withdraw and stop initiating behavior or reacting in the same way that they did before. And unfortunately, at the moment, there really is no agreed treatment approach for apathy. Some people who go to the clinic and say they're not feeling very motivated get given antidepressants. But unfortunately, antidepressants can actually make apathy worse. And that's one of the things that I'll talk about a bit later in the talk, showing some of our data. So there's really not much of a sort of an agreed treatment approach. And this could be in part driven by a lack of animal models that really tap into the motivated disruption that we see in apathy patients. But of course, patients don't really go to the clinic and complain of a reduction in goal-directed behavior. They complain of a reduction in motivation defined by things that make up activities of daily life. So things like going out for a meal with friends, seeing friends, doing hobbies, playing with your pet, seeing your family, traveling, having hopes and dreams, working, these kind of things, these internally generated behaviors are the things that become disrupted in apathy and really sort of become defined by this overwhelming sense of basically, I just can't be bothered. And actually, I like to kind of go back and think about the patient experience by looking at patient transcripts of patients with apathy talking to clinicians to really get a good idea of the patient experience of apathy. And here is an example of a doctor talking to a patient who's had a stroke actually and has got apathy as a result of that. And the doctor's asking, well, what would we have seen if we'd known you a few years back? And the patient's saying, oh, I'd be buzzing around, being busy all the time, but I've just slowed down and more or less come to a halt. And I don't really know what's stopping me. So it's really that change in behavior where you used to be a busy, active person and now you're withdrawn. You don't want to go out and do the things you used to want to do, but you can't really explain why. So that brings me on to how we sort of typically measure motivation in animal models, because of course, animal models can tell us more about things like the underlying neurobiology of these disrupted kind of processes. We can test treatments and understand behavior in a bit more detail. And one of the more commonly used paradigms is called the effort for reward task. So this is what's known as an operant task. So the rodent is placed in an operant box and they learn to make an effortful number of responses for a high value reward. So that could be something like nose poking in an aperture, pressing a lever, touching a touch screen. And from that, they receive a high value reward. So this is something like some kind of tasty food, a reward pellet or something like that. Or instead of exerting effort, they can choose to consume freely available but pretty boring lab chow. And the decisions that the rodent makes across this session gives us a readout of their motivational state. So if they choose to do more of the effortful responses, we can say they're in a higher motivational state. And if they shift towards the lower motivation option, then we can say they're in a lower motivational state. And these kind of tasks have been used for years now to really start to get insight into how the dopaminergic system interacts with motivated behavior and how it drives it, and how we can start to kind of think about different therapeutic targets relating to that system. However, there are some caveats with using tasks like these. Even though we get a lot of control and we can kind of decide when the animal is exerting that behavior, and we can kind of look really closely at the number of trials they're doing, there are a few things that kind of lead to us guessing that. And that's requiring things like food restriction to induce a robust behavioral response, and having to use a really simplified test environment to get the animal to actually do what you want them to do in the time that you want them to do it in. And because the animal is sort of already put into this sort of motivated state by taking that food away and asking them to learn to respond for food, and kind of taking away every other behavioral option by putting them in the simplified testing environment, it really got me thinking about how well do these kind of tasks align with the motivational deficit that's experienced by patients with apathy, which is more about this disruption to this inner drive and not something to do with kind of working to get something external. So to kind of address this, I decided to try and develop a new task that I felt kind of better aligned with these intrinsic motivational processes and a way of looking at motivated behavior without having to food restrict or train the animal. So to kind of really tap into a more naturalistic behavior and a more sort of innate drive. So I developed the effort-based forage task. So here's a picture of it here. So it's comprised of two sort of main compartments. We have this sort of safe and enclosed compartment here where the rodent has access to everything it needs to sort of live more or less happily. So it'll have access to food and water and wood chip bedding and shelter. And it can choose throughout a 2-hour session to traverse through a tube into what we call the foraging area, which is perceived as more open because it's got a barred lid at the top. So it experiences airflow differently from when it's in a safe and enclosed environment. And the rodent can essentially choose to forage for nesting material. So it exerts effort to pull nesting material from this 3D printed and custom designed nesting material box. And the amount of nesting material that's shuttled gives us a readout of motivational state. So I've got a video of the mouse doing this behavior here. So because this is such a innate or intrinsic behavior, we don't actually have to train the mice to do this. They just instinctively know what to do. So while we do habituate the animal to the arena over a few sessions and establish a stable baseline, the animal knows what to do. So we don't actually have to go through those long training periods that we have to do with operant tasks. So for example, in the effort for reward task I just mentioned, we'd have to probably wait about 4 weeks before we get the animal to a point where we can start doing things like, testing different manipulations to understand motivated behavior better. Here, the animal starts performing almost immediately. And once they've overcome that initial sort of anxiety of being in the arena, we get that robust output straight away. And that remains stable, so we can do lots of different manipulations across time. So that's a sort of basic setup of the forage task, but we can also do sort of by changing different aspects of the task setup, we can start to tap into different sort of motivational processes or behavioral processes. So in its sort of standard form, we kind of get this overall idea of motivated behavior, but there could be lots of different things that sort of impact on the foraging behavior that lead to that kind of overall conclusion of a motivational deficit or a motivational increase. So one of the things we can do is actually change the amount of effort required to forage from the nesting material box by changing the aperture size of the bedding box faceplate. So we can see it here. So these are the apertures through which the animal forages, and we can basically change the size of that to kind of make it more or less difficult and see how the animal responds to that. So we call that effort-based modulation of behavior. We can also look at the animal's response to a more aversive environment. So we know that mice don't really like open spaces. So one way of testing how an animal responds to a stressful environment, or I like to call it affective reactivity, is by making the foraging area slightly larger. So the animal is less likely to want to forage for a long period of time inside that more open area because they feel a bit more exposed. And so we can look at not just effort-based modulation of behavior, but also their affective reactivity. So how strongly are they responding to a more aversive environment, and how does that also impact on foraging behavior? So by kind of tweaking aspects of this task setup, we can look at lots of different types of behavior. And I do think now, we're moving towards behavioral batteries, which I think is a great idea. We can't really make these grand conclusions based on, one output. And so this is a way of almost kind of doing a behavioral battery, but not really changing the context of the task. So we can ask the animals to perform different behaviors within that same task context. So you stop getting some of that variability by having to use lots of different tasks to ask lots of different questions. And kind of bringing this back to apathy then, we're not just looking at things like just general motivation. We can start to tap into these more specific domains. So with the effort curve and with this kind of general foraging paradigm, we can look at behavior cognition, so reduce effort to start or finish a task, and then emotional affective as well by looking at affective reactivity. So have they stopped responding to an aversive environment, and does that give us an indication of emotional blunting? I just kind of want to touch briefly on, how does species-specific behavior actually fit into the context of translation? And I think this is something that definitely up for discussing after the talk. And in what we really think translation is, because of course we're always trying to translate our animal behavior to human behavior. Of course, that's kind of what the whole point of this thing is. And I think some people like to use tasks that have things like touchscreens, for example, because humans use touchscreens. And if humans use touchscreens and animals use touchscreens, that's a way of achieving translation because our behaviors are very similar. And because of that, maybe that means we're tapping into similar brain circuits. And I do wonder then if that's why we started moving towards that kind of thing, because if we can relate an animal's behavior to our own, is it easier to interpret? And I think as a society, we like to relate animals' behaviors to our own. There's lots of films and media based off of it, of animals acting like humans. But do we really need to do this always in the context of behavioral neuroscience? And really, as long as the tasks that we use tap into the same behavioral domain as a human, I think we can achieve translation without necessarily having to ask them to perform the same behaviors. So we can ask a mouse to perform like a mouse, and we can ask a human to perform like a human. But as long as we're tapping into the same shared behavioral domain, then that's where I think we can achieve translation. Okay, so before I start to kind of go into the what we found with the effort-based forage task, I'm just going to briefly touch on what the task output actually looks like. So when you see a bunch of graphs that I'm showing with things like total nesting material shuttled and percentage nesting material shuttled, you know what I'm talking about. So one of the outputs that we use a lot is total nesting material shuttled. And this is where the mouse has foraged nesting material from the forage box here. They've put it and they've taken it and shuttled it back to their safe and enclosed area here. The task is 2 hours long, so we're not looking or assessing nest building behavior. We're really capturing that initial foraging sort of activity. So total nesting material shuttled is how much they've taken out of the box and taken back to their house. Percentage nesting material shuttled is a slightly different way of looking at that. It's a way of integrating how much they've foraged with how much they've actually taken back. So in some phenotypes and under some drug treatments, some mice will forage from the nesting material box, but leave it just out in the open here, or only take a proportion of it back to their home area. And we think this is actually a really interesting behavior that might relate a bit more to their sort of stress-related behaviors, and I'll touch on that later. So when I'm presenting the data in this talk, I'm really looking at their foraging and taking back to the home area, and that expressed as a percentage. So how much have they actually taken or left behind? Okay, so when I'm skipping a lot of time here. We set up the task. We had to figure out the times, how to actually give them the nesting material, what's the best kind of nesting material, etc., etc. So I'm skipping all the way to, okay, we've done all that, and now we're validating the task. And of course, the kind of classic way of validating a motivation task is to modulate dopamine in some way, because we know that dopamine is really commonly involved in motivated behavior. So one of the first things we did was some acute pharmacological manipulations. So one of the treatments that we first used was haloperidol. So haloperidol is actually mainly known for being used as an antipsychotic in the treatment of things like schizophrenia. But for us, it was really useful because it's a D2 antagonist. So it reduces the amount of dopamine in the system. So we can look at what happens when we lower dopamine in the animal, and does that impact on foraging? And we found that the higher dose of haloperidol, which actually aligns with the clinically used dose for those used in schizophrenia, reduce the amount of foraging in the task. But you have to be careful when you're modulating dopamine, because dopamine is also really involved in movement. So we had to kind of add in a behavioral control there. I haven't shown this, but essentially we had a passive infrared sensor above the foraging area. So we could look at their general activity and see if that changed for drug treatment. And we found that while general activity wasn't changed, their actual foraging was. So we could be a bit more confident that this isn't impacting their motor performance, but is more specific to their foraging motivation. But you can see here we have quite an unusual distribution of data. And there's a sort of what I'd call quite a significant spread. So we kind of wanted to look at that in a bit more detail. By separating the mice out into lower motivated animals and higher motivated animals, nothing fancy, literally just a median split. And we found that mice that were in a lower motivated state were more sensitive to the haloperidol treatment than those that were in the higher motivated state. So it kind of suggested that this task is sensitive to just sort of differences in basal levels of circulating dopamine. And then we could see that interacting with our drug effect, which is really interesting. So on the flip side of that, we also wanted to ask, okay, well, what happens if we increase dopamine? Will we also increase motivation? So we used a drug called methylphenidate, and this is typically used to increase dopamine in patients with ADHD, for example. And we found that increasing methylphenidate increased foraging behavior without having any impact on their general activity. And again, by separating the animals out into their lower and higher motivated groups, we found that in lower motivated animals, they were more sensitive to the lower doses of methylphenidate, whereas in the higher motivated group, it was only that largest dose that had an effect. So again, we're seeing sensitivity to those sort of basal levels of dopamine and how that interacts with the drug effect, which is really exciting. And I think, you know, when we're using operant tasks and we're food restricting and we're getting all of our animals into a really high motivated state, we end up getting really nice tight error bars. And I know tight error bars are a thing that people like, but I think we do start to lose that sort of individual variability by kind of pushing all the animals towards a mean. And by not having this sort of restricted state and the animals just acting in their sort of intrinsic drive, we start to see that spread of motivational state, and we can start to see differences in drug effect like this. So that kind of at the very basic level, we found a drop in dopamine drops motivation and an increase in dopamine increases motivation. So we could be a bit more reassured that we are tapping into a dopamine relevant process here that looks a bit like what we've seen before in terms of motivated behavior. Okay, so moving on then to using phenotypic models in the task. So one of the first ones we looked at were aging. So aging is associated with a change in motivation over time. So we see a decline in motivation and in some patients or some older people, an emergence of apathy. So I was very interested in motivational changes in aging, and I wanted to see what they looked like in this task. So I actually tried some of the variations of the task that I mentioned. So what we call the effort curve paradigm here, and also the affective reactivity paradigm, which I'll discuss in a second. So you can see here in this graph, in the younger animals, we see a really nice effort-based modulation. So they're foraging more when the aperture is easier than when the aperture is really difficult. They're foraging less. So we see this really nice line here. But in the aged animals, we see a sort of blunted effect. They're not modulating their behavior relative to the effort. We're seeing sort of a difference in the shape of the curve. Now, it's possible that the animals could have sort of physical impairments that make it more difficult for aged animals to forage their nesting material. So we actually doubled the amount of time that the animals are allowed to forage for to account for the fact that the aged animals might be slower. And we actually used aged mice with a bit of a caveat in that they weren't super old. They were about, I'd say, 15 months old. So they hadn't really got to that point where they start to get all these sort of nasty age-related effects that mice get when they're older. So they were in good physical condition. But we can still see this sort of blunted effort-based modulation of behavior, but only when the aperture was actually at its easiest. So they should have had the easier time there. So we can say that these aged animals have some kind of blunting in their effort-based modulation of behavior. But what about their affective reactivity? So here I'm showing the shuttling output measure that I talked about before, and also the percentage nesting material shuttled. So first of all, let's look at the total nesting material shuttled. So we have the young animals and the old animals both forage less when the foraging box is placed in a larger environment. And that makes sense. They're suppressing their foraging behavior because it's an aversive environment. But when we look at the percentage of nesting material shuttled, we see that young animals actually shuttle less than of what they forage when they're in a larger area. So they're taking it out, but they're leaving it there and running away. And we think that might be because when they're in a more exposed environment or when they're feeling more stressed, they feel more vulnerable when they're carrying things in their mouths. They might run in, forage a bit, drop it and leave. And that's why you start to get this sort of build out in the open rather than shuttling it through. But in aged animals, we don't actually see this. They stay pretty much the same. So it looks like a sort of blunting effect where they're not really changing their percentage shuttled behavior when they're in a more aversive environment. So it looks more aligned to something like an emotional blunting. But when we contrast that with court-treated animals, we start to see something different. So chronic corticosteroid is used as a sort of model of depression-like behavior in mice. So it's the corticosteroid is the stress hormone in mice, kind of analogous to cortisol in humans. And again, we did the effort curve here. And it looks a little bit different to the aging profile, where we just see a sort of a drop in the court-treated animals. So their overall foraging level is just basically lower than the kind of healthy controls. So we're not seeing really a change in the shape of their of that curve, just a sort of overall lower amount. So they haven't really changed their effort-based modulation of behavior. They just have a sort of like lower sort of motivational state. And when we look at, again, this affective reactivity, as we would expect, we see that their foraging is suppressed in a larger environment. But this time, the court-treated animals have an exaggerated change in their percentage nesting material shuttled. So they have a greater decrease in that percentage nesting material shuttled, which means they're leaving more than the healthy controls out in the open in that condition. So it's more of they're sort of in a more exaggerated stress state. So by using the same combination of variations of the task, we can kind of conclude that aging has more of an apathy-like profile where they're showing something akin to a change in effort-based modulation of behavior and an emotional blunting, whereas in court-treated animals, we see an overall reduction in motivation and an exaggerated stress response, which is more akin to a depression-like phenotype. So by changing bits of the task, we can actually start to pass different behavioral conclusions from animal models, which have quite similar behavioral profiles. And the next thing I wanted to really ask was, okay, what are we actually measuring here? Are we looking at the same kind of motivated behavior that we see in an operant task, but just with the added advantage that we don't really have to train them and we don't have to food restrict them? Or is this type of motivation different to what we've seen before? So one of the first questions I asked was, okay, can we devalue the reward in some way? So people in the motivation field kind of say, if an animal's doing a goal-directed behavior, you can devalue the reward. So, for example, in the context of an operant task, you could pre-feed them. So the food isn't actually that rewarding anymore. So they're going to be less willing to put in the effort to get it. And doing that with nesting material is a little bit more tricky because we can't do something like pre-feed them, but we can kind of think about why they're doing the behavior and see if we take that away, does that behavior still persist? So one of them was, okay, well, what if they're just foraging for nesting material because they have no other option? They don't have any nesting material and they need it. So I put just a bunch of free access to nesting material in their home area. So they didn't have to make any kind of effort whatsoever to get that nesting material. And they still foraged the same amount as they did when they had no nesting material. So they're not foraging because they don't have anything. So that was number one. And the other kind of explanation is that the mice are cold. So there's this idea that mice in laboratory conditions are actually quite cold. We don't keep them in high enough temperatures, and actually their thermoneutral zone is around 27 to 30 degrees, but we're keeping them in lower temperatures. So what if they're foraging for nesting material because they're cold and they need it? So rather than it being they're hungry and they need food, what if it's they're cold and they need nesting material? So we warmed up the test environment to kind of take that aspect away. And rather than reducing foraging, it actually increased it. So warming the environment and providing free bedding for the mice didn't stop that foraging behavior. So it wasn't the kind of goal-directed behavior that we typically see in an operant kind of setup. It's almost like the act of foraging itself holds a reward. So the act of foraging itself is intrinsically rewarding. And that got me thinking about our sort of typical view of motivation and reward and how we design tasks to measure it. So the kind of typical view is we do the thing, we do an action, we receive a reward or we avoid a punishment, and then we remember it. And because we remember it, we do it again. And that happens over and over again. And in animals and humans, we get this nice learning curve and eventually it stabilizes out. And that fits kind of into how we measure motivation. So if we do the thing more, we say, oh, they're more motivated. And if we do it less, we say they're less motivated. And that fits very neatly into the kind of operant paradigms where, you know, the animal learns to do something like press a lever, which is doing the action. They get a tasty pellet, which is receiving their reward. They remember that association and they do it again. But how does that apply to foraging? It doesn't. They do an action, they don't receive anything for it because it doesn't seem like actually getting the nesting material is the reward. They don't remember it because they never had to learn it. It was there in the first place. They just do it and they do it again. So it doesn't really fit that typical view of motivation and reward, which I think is really interesting. But it seems like the action itself is the thing that's rewarding. And I think the task is allowing us to tap into a slightly different dimension of motivation to what our typical tasks do. We're able to look into things like intrinsic motivation. And as I said before, when a patient goes to a clinic, they're not really talking about their goal-directed behavior. They're talking about things that are internally rewarding, doing hobbies, seeing family, traveling, that kind of thing. So this task seems to align more nicely with that sort of difference in intrinsic drive. Okay, so I'll just do a first summary here. So the effort-based forage task provides a readout of motivational state that's sensitive to dopaminergic modulation and phenotypic models. And we can use variations of the task to start to pull apart distinct disease profiles. And it seems like foraging is rewarding and reflects an intrinsic motivation. Okay, but why does that actually matter? So examining motivation purely in the context of extrinsic reinforcement means that when we try and treat it, we might actually be missing a whole behavioral domain that is really critical to the patient experience. And it might actually explain why so many of our first line treatments for treating motivation loss don't really work very well or can actually make it worse. So I'm thinking things like antidepressants. And even things like I said, with the antipsychotics, haloperidol in the context of schizophrenia may treat things like psychosis, but can actually make their motivational deficit a lot worse. And we miss a lot of information about how motivation might shift or change with disease. So what if extrinsic motivation shifts or intrinsic motivation shifts towards an extrinsically motivated behavior within a disease context? And of course, we use behavioral tests to start to think about neurobiology and model things computationally. But if we're only using a sort of set type of behavior, we could be missing a whole bunch of different things. And it seems then that when we think about motivation in the context of research, we really are focusing on that extrinsic motivation because it's easy to quantify. An animal does something, gets something, and we're able to measure that. But it might be that in terms of symptom experience, there's a sort of more of a balance of extrinsic and intrinsic motivation that we're not really tapping into at the moment. Okay, so as I mentioned, antidepressant treatment is a bit of an interesting one. So people go to the clinic saying they're not very motivated, and more often than not, the clinician will assume that they're in a depressed state and prescribe antidepressants. But in a certain patient populations, that may not be the case. They may be suffering from apathy. And in this case, treatment with SSRIs in the clinic can actually make it worse. And I was interested in how these two different tasks differ in terms of their sensitivity to a whole bunch of different antidepressants. So selective serotonin reuptake inhibitors, and other kind of types, so multimodal antidepressants and noradrenaline reuptake inhibitors as well. But I won't go into all of that there, but just to say that we do have a paper out on that if anyone's interested to look in a bit more detail. So first of all, and this is just acute treatment, by the way, we did also do chronic treatment, but again, there's quite a lot to go through and I didn't want to sort of overload it into this talk, but again, that's all in the paper as well. So this is acute treatment, and we trained our mice to actually voluntarily take the drug in a palatable solution. So we were trying to get it as close to the clinical experience as possible. We used clinically relevant doses adjusted for animals, and we trained our animals to take it in condensed milk. So they were consuming it orally just as a patient would. And we can see that in the operant effort for reward task, treatment with escitalopram, which is an SSRI, actually increased the number of effortful trials that the animal was doing, which suggests that the animal was in a more highly motivated state with treatment with escitalopram. However, when we did the exact same thing, using the exact same doses, exact same route of administration, and so on, we saw the opposite effect. So escitalopram decreased motivation in the effort-based forage task. So we saw a really profound divergence in the behavioral response, the same drug, in two tasks that theoretically measure motivation, but in very different ways. So we tried a bunch of other different antidepressants to kind of see if this was a kind of consistent thing. And again, we saw the same thing, an increase in motivation measured by the effort for reward task, but a decrease in motivation measured by the effort-based forage task. So this is really important because, you know, these kind of tasks are what we kind of base our kind of interpretation of drug effects on. And this task is telling us that antidepressants increase motivation acutely, but the effort-based forage task is telling us the opposite. They actually decrease motivation. So this is really important because we're making very different conclusions based off two different tests. So one kind of explanation for that could be that there are differences in the task environment that are driving this divergent effect. So there's a lot of key differences in the effort for reward task. It's a single sort of task environment. They're hungry, whereas in the effort-based forage task, they're not hungry. There's two different compartments. So the setup of the task is quite different. And the effort-based foraging task has that potential to be sensitive to anxiety because the foraging area is out in a separate part to their safe and enclosed area. So they have to sort of go out into the open to obtain that nesting material. So you get that added kind of like, which is also a strength of the task when you're interested in anxiety, but anxiety could be coming into play here. because some people have found that acute treatment with SSRIs actually does increase anxiety in some people. So to kind of account for that, I developed a new task called the dual effort choice task. So here the animal has two options. They have that safe enclosed area like the effort-based forage task, but here they can either choose to go into the tube and forage their nesting material, or they can go into another tube and nose poke to receive a tasty reward pellet through a pellet dispenser called a Fed3. I don't know if anyone's familiar with that, but they're really useful. You're able to kind of do operant-based behaviors outside of the context of an operant box. So we utilize that here in the dual effort choice task. And interestingly, we found that this foraging deficit is consistent in the dual effort choice task. And whereas the pellet behavior is slightly different. So let's look at escitalopram first. So here we can see that in the dual effort choice task environment, we see that reduction in foraging as we observed before in the effort-based forage task. And we see an increase in the number of trials on the Fed3 device as we did in the effort for reward task. So that divergence is conserved when you make everything equal and the same in the dual effort choice task. And again, looking at both sertraline and venlafaxine, we see a decrease in foraging with treatment with these antidepressants, and we see no effect on the pellets. So we could be confident that we weren't just seeing a deficit in foraging because they were anxious and not going out, because they're going out to get pellets, and that's not really impacted. So we see that in this task context, foraging is consistently reduced, but trials is a bit different. It depends on the antidepressant you use. And we can be more confident then that these observed differences we see between the two tasks aren't just driven by something like anxiety. So second summary then, acute SSRI treatment induces opposing behavioural effects on the effort for reward task and the effort-based forage task. The choice of behavioural tasks can lead to profoundly different conclusions about drug effect, which suggests that differences between motivation type has quite significant translational implications, and it's certainly something that I think we need to be aware of. And the use of the dual effort choice task highlights that simple differences in task environment do not explain that drug divergence. We can be a bit more confident that these drug effects are really specific to motivational type, not just down to task environment. Okay, so you've got to ask though, what else is influencing behavior in the effort-based forage task? We can say we're looking at motivation, but we're using a more complex environment here, and it's tricky to kind of isolate a pure behavioral domain like pure motivation. And actually, I do think this is more of a strength for translation, because as we behave out in the real world, we don't have these sort of pure behaviors that we are measured or experienced. But it's important that we understand what other kind of things are going on that are impacting on the motivation in the task. And it's important as well for other aspects of neuroscience and pharmacology and beyond, because we use behavioral tasks as our sort of fundamental building block. We build conclusions about neural circuits and therapeutics based off of our choice of behavioral task. So we really need to make the effort to really understand the behavior that's underlying the motivation that we're looking at, and really get to the question of what is this behavior telling me before we start to use our fancy techniques that go into the nitty-gritty of things. So I kind of thought a bit about how to do this, and as someone who has a pharmacology background, the main thing that's always suggested to me first is, what about more drugs? And they can be useful, but I'm more interested in things like sort of endogenous behavioral variation and what that could tell us about the performance of the task and what kind of things might be impacting on motivation in that way. So one thing I thought about was using strain differences to actually understand foraging behavior a bit differently. So people use lots of different strains in neuroscience and beyond, and I think we can all kind of generally agree that different strains have very different behavioral profiles, and we can actually use that to understand the foraging behavior in a bit more detail without having to do something like an acute pharmacological manipulation or use a disease model where there's lots of other different things going on. We can use sort of natural variation that already exists out there. So I used 3 different strains, and I should have said before, actually, all of this stuff up to date has been in C57 black mice. So we used those as before, and we also use 129SV mice and bulb C mice. So some people might have experience with those strains, but the bulb C mice and 129SV mice are generally associated with higher levels of anxiety. 129 mice are actually interesting. Some people think they might be a useful model for schizophrenia because they have a little genetic mutation in their DISC1 gene, I believe, which makes them have lower levels of cognition. Bulb C mice are very impulsive, and actually have been shown to be really good at operant tasks because they're so impulsive and like able to fixate on certain behaviors. So because each of those mice have a really interesting but very different behavioral profile, it can provide insight into the behavioral dimensions that are actually impacting on foraging behavior in this task. So the first thing we did was basically just ask, okay, when we habituate them to the task arena, what does their activity look like when they're in the foraging area before we even start to add foraging into the equation? And we can see that while C57 mice sort of kind of habituate to that task environment by kind of reducing their activity over time, with the more anxious strains, 129 and bulb C, we see actually an increase of activity in the foraging area with sessions as they become a bit more confident. And bulb C mice are definitely the most active in that foraging area. So when we moved on to looking at their foraging levels, I had it in my head that bulb C were going to be these amazing foragers, and the 129s who had lower levels of activity just generally in the foraging area weren't going to be very good. But I was completely wrong. And actually, the 129 mice were the best foragers of the lot, and the bulb C mice were actually really low at foraging. And in fact, it was really difficult to get an effort curve from them because they just weren't foraging that much at all. So we didn't need like acute pharmacology or disease models to see that the task was sensitive to strain-based differences in foraging behavior, with the largest differences between the 129 mice and the bulb C mice. But again, as with the SSRIs, you could argue, well, this could be down to anxiety in some way, and we already know that those mice have differences in anxiety relative to the C57 mice that might be relevant here. So we use two different types of anxiety tests that I'm sure people might be familiar with, but just in case, we use the elevated plus maze, which basically kind of gives you an idea of anxiety by seeing how much time the mouse spends in the open versus the enclosed arms. So a mouse prefers to spend more time in the enclosed arms because that's where they feel safer. But a greater time spent out in the open indicates a lower level of anxiety. And 129 SV mice spent a lower amount of time in the open arms expressed as a percentage of their total activity relative to C57 mice. So in this context, the 129 SV mice were the more anxious strain. And in the novelty suppressed feeding test, here we're looking at the mouse kind of doing a cost benefit analysis. So they're put in a large kind of open space with food in the center. They're really hungry, so they want to go and eat the food, but that kind of contrasts with their sort of innate kind of behavior of wanting to stick to the side. So it's whether the mouse can overcome that and eat from the middle. And the faster they do that, the less anxious they are. And actually, the bulb C mice trended towards being the least anxious by having a lower latency to eat. So what we're looking at here is actually generally the 129 SV mice are the more anxious strain. The bulb C mice are trending towards being less anxious. So the fact that the bulb C mice forage the least and the 129 mice forage the most suggests that this difference in foraging is not explained by anxiety, or at least traditional tests of anxiety. So that's really useful information to know as well. So in foraging, the 129 mice are coming in first place, and the bulb C mice are coming in last. But what if we use the effort for reward task, so that operant task? And it's the other way around again. So the bulb C mice actually perform the best compared to the other strains. So this time it's flipped round. The 129 mice were actually performed the lowest and actually showed a lot more behavior towards the low effort, low reward option, which I haven't shown here. So again, like with the SSRIs, we're seeing a complete inversion of our conclusions about a motivational phenotype within these strains. So it's not just pharmacology where we're seeing a divergence here, we're seeing it on a phenotypic level as well. So again, it really shows that intrinsic motivation and extrinsic motivation are two very different components of motivation and can have really opposing conclusions based on the task that you choose. So my next steps really are to kind of figure out what's going on in the brain. Like we've kind of built up our whole understanding of motivation based on extrinsic motivation. So does dopamine behave in foraging like we would expect in a more classic operant-based task? And to do that, I'm using neural recording with fiber photometry. So I've got a little, hopefully video works here. So this is a mouse that's tethered whilst foraging for nesting material. So it's a freely moving behavior. They're completely unimpeded. And we've developed some habituation protocols so they're not stressed by the tethering or the task context. And we can start to get insight into the real-time signaling of dopamine in the nucleus accumbens while they're doing this behavior. It's quite tricky because freely moving behavior is very chaotic, and we're not doing a trial-based task here. It's all freely moving. So I'm sat there watching videos and scoring exactly when they forage from the nesting material box, aligning those with the fiber photometry signal and seeing what we can get. So it's very early on now. But interestingly, we're seeing things like a peak just before they actually start to forage, which is very different to extrinsically motivated behaviors, where we see a peak at at the event itself, like where they're consuming a food reward, for example. Whereas here it seems to be shifted back. And we'd only really ever see this in a queue-based task where they've learned over time that a queue predicts reward. But of course they haven't here. They've never really learned this before. So there's already a very interesting difference in dopamine profile that warrants further investigation. But foraging isn't just for disease phenotyping and pharmacology. We're extending it out into lots of other different fields as well. So, for example, looking at it to look at things like changes in circadian rhythms of behavior. We're using it in some computational modeling of intrinsic motivation at the moment, because, of course, this type of motivation really does break down all of the sort of reinforcement learning models that are kind of out there at the moment. It doesn't really follow that same pattern. So we're trying to come up with some new computational models to account for this behavior. And we've also found that because the animals' foraging levels are so sensitive to stress, we can actually use it as a welfare readout as well. So we can ask questions about different protocols. Are they stressful for mice? Or we can put them in the forage area and ask that question by looking at their foraging levels. So it's a task that's useful across lots of different fields and kind of help get that out there. We started to kind of put things out there like 3D models, renders of the task, and kind of put everything open access so that people can start to use this task themselves. And of course, I'm happy to always chat about it. I love talking about foraging, and I'm always keen to help people get this set up in their own labs. So do get in touch either through the forum or through my email if you're interested in setting that up. But the great thing about it is the animals don't really need training. They know exactly what to do. So as long as the test conditions are all okay, it should work without having to put in too much time, experimental time. So I'd just like to finish off with some acknowledgements. So thank you to Professor Emma Robinson for providing that research environment, but definitely to all the amazing students that helped out with all the various different projects going on. I couldn't have done it without them. And thank you as well to all the various sort of funders that kind of helped fund this project. So thank you for listening, and I welcome any questions.
Thank you, Megan. This was an extremely interesting presentation, and I think kind of very sensibly investigated test, which is at the start a very elegant paradigm.